Yeah, thank you very much. Yes, I'm Donjete Simnica, and I'm a fourth-year PhD student at the lab of Masha Binder at the University Clinic in Halle, which is a really small town close to Berlin in Germany. And today I will talk about our findings on the T-cell receptor sequencing of the samples that we received from patients with COVID-19. And this data set was published just recently in Immunity, and I'm going to talk just about the T-cell receptor sequencing now. And probably just as many of you who are listening right now, we are also interested in understanding why some individuals clear the virus much better and only with mild symptoms, and others develop severe disease. So we started collecting samples in early April from hospitalized patients in our hospital as well as a collaborating hospital in Frankfurt. And we mobilized recovered individuals all over Germany to participate in the study and documented their symptoms with a questionnaire. The only inclusion criteria for this was a positive PCR for SARS-CoV-2. And here you can see that the two cohorts that we collected during this time, one of our cohorts consisted of 17 patients at recovery, which from mostly mild COVID. This is cohort 1, and we think that probably through a fast and efficient clearance of the virus. And then the second cohort consists of 18 patients with active disease who developed very severe disease with the need for example, mechanical ventilation or extracorporeal membrane oxygenation, a very difficult word, and presumably through inefficient viral clearance or a deregulated immune response in some way. Moreover, we had two, let's say, index cases or interesting cases from whom we had samples during active disease as well as recovery, or rare cases. Approximately 60% of our patients were male, and the two cohorts also differed at the median age. So the cohort 2 was older than cohort 1, which is a little bit in line with the knowledge that we have now of patients who are more at risk to develop severe disease. So we performed bulk sequencing of the B and T cell receptor repertoire from the DNA of, at that time point, 69 samples that were available to us, and deposited all the data, the repertoire data, as well as the annotated data according to the Meyer standards at the I-receptor gateway under this study ID, which you can see here. And yeah, as I mentioned before, I will focus on the T-cell receptor data today. So just for some of you who maybe don't have quite in mind what the T-cell receptor repertoire is, so each T-cell has an antigen receptor, which is depicted here, which can bind to antigens that are presented on MHC molecules. And this receptor consists of two polypeptide chains, and in the majority of cells, these two chains are alpha and beta. And these polypeptide chains are encoded by gene fragments, which are called B, D, or J, and in a random fashion in a process called BDJ recombination. And then at the genomic DNA level, we end up having these rearranged gene loci, which we can sequence. In our approach, we sequence the beta chain of the T-cell receptor. So we approached our data in different ways in order to understand what's happening in the repertoire, in the successful versus failed immune response. And first, we calculated general T-cell repertoire metrics like diversity, clonality, and the VJ gene usage, for example, since these are really reliable parameters of an ongoing immune response, as we have seen in earlier studies that we've done. So the repertoire diversity here on the left, depicted just as a little cartoon, can be estimated through the richness and the evenness of the clonal distribution. And the richness would be the number of unique clonotypes found in the repertoire. The evenness would be the abundance of the clonotype in the repertoire, or the distribution of the frequency of that clone within the repertoire. So a repertoire with a really high number of unique T-cell clones and a rather even distribution of these clones would show a really high diversity, and a repertoire with the low number of unique clones and, let's say, very dominant clones would show a low diversity. Next, we hypothesized that the samples of the recovered COVID-19 patients should still have the clones at a detectable level, which are directed against the virus. So, and which also help to successfully clear this virus. So, since there's not this one anti-SARS-CoV-2 clone, we used the tool Glyph to group lymphocytes according or presume to their potential, let's say, antigen recognition. So with this, we hope to identify, TCR specificity groups within all of these different T cells that we sequence. And finally, we adopted the approach of the longitudinal monitoring of clonal trajectories that Ms. Walczak was just presenting before me from the paper of Minervina et al. And with this approach, we want to identify groups of clones that present similar dynamics during the course of disease. For example, an expansion pattern towards recovery, which biologically would mean that somehow these clones are being selected or expanded and are used for the individual to recover from the infection. So here you can see the repertoire metrics. We have also used repertoires of 39 age-matched healthy donors as a reference, here depicted in blue. And what was interesting was that we found that individuals at recovery had a really rich and diverse T-cell repertoire, even above the repertoire or the levels of the healthy donors. This was quite in line with the overshooting T-cell regeneration that we also measured by flow cytometry in a subgroup of patients, which you can see here. Patients with active and mostly severe disease had a rather clonal repertoire as compared to the other two cohorts. So we then went further on to look at the combinatorial VJ gene usage, and what we saw was that patients with COVID, either at recovery or at active disease, which is this, which are these dots here, really separate from the repertoires of healthy donors. And when we then looked at single B gene usage or J gene usage, we saw that actually at active disease we have an overrepresentation of these B genes. And we have to keep in mind that active disease in our cohort also means severe disease in most cases. And at recovery, we saw an overrepresentation of these B genes. The J gene usage was rather uniform, or let's say uniformly distributed. So then I will continue with our Glyph results. So this is quite a busy slide, but I will walk you right through it. So what you can see here is we grouped the T-cell clones using the Glyph tool to clusters. And on the x-axis, you can see the frequency of the clusters, and all dots that go to the left are enriched in patients with active disease, and all dots that go to the right are enriched in patients after recovery. And the size of the dots then represents the number of individuals in whom the cluster was found. We also calculated the generation probability for each cluster, and this is plotted here on the y-axis. And keep in mind that the y-axis is inverted, so it means that the low generation probability is on top, and clones or clusters with a high generation probability are plotted on the bottom. And for you who don't really know what to do with the word generation probability, so this in general would indicate, the generation probability indicates for each VDJ recombination of how probable it would be that it is generated through random VDJ recombination. So there are some VDJ recombinations that are more common, and there are some that are less common. The ones that are more common would have a high generation probability, and we would expect to see them in a big proportion of the population. And the ones that are less common of being generated would have a low generation probability, and we would not expect them to be shared. So here in the gray area, yeah, maybe that's also interesting for you to know, we put our threshold for the low and high generation probability. So in total, we found a little bit more than 16,000 T-cell clusters, which were, and most of them were shared between the two cohorts. These are depicted here by the open circles. And oh yeah, maybe one more thing. Our threshold for calling a cluster was that it has to contain at least 4 unique T-cell clones, and it has to be shared at least between 3 individuals. With this, we wanted to make sure that we don't have so much noise in this analysis. So it is worth noticing actually that the majority of these shared clusters that are also public are enriched in the active disease and severe disease patients. And the filled dots represent clusters that are only present either in the active disease, which are here, or at recovery, which are much more. However, what we also found was really interesting were some clones here in the gray area, which are low generation probability clones that are shared only by patients at recovery, which you would not expect to have them shared amongst so many people. And these are around 31 clusters, which is not very obvious here, but these are 31. And since they're shared between so many recovered patients and have a very low generation probability, we hypothesize that these have something to do with SARS-CoV-2 infection and have been selected probably in the repertoires of these patients during this infection. Okay, and the third approach to try to make sense of these repertoire data was the longitudinal analysis of the clonal trajectory. And in the immunity paper, we had only the longitudinal samples of our index patient 7, from whom we had samples during active disease as well as recovery. And this patient had a mild disease course, indicating that he or she coped with the SARS-CoV infection successfully. So first we clustered the frequency dynamics of the top 1,000 clonotypes and identified 4 distinct patterns of these dynamics, let's say, and plotted them in this principal component analysis on the left. And the majority of clonotypes belong to pattern 1 or 2, which are shown here by the yellow or the blue dots. And they quite nice, separate nicely here on the principal component 1. So therefore, we focus only on these 2 patterns and plotted the trajectories of these 2 here on the right. And what we saw was that actually clones belonging to pattern 2 showed a rather contracting trajectory or dynamics, and clones belonging to pattern 1 expanded towards recovery from the disease. We also looked then for the clones that belong to pattern 1 in our clusters that we found with Glyph. And we found that some of these clones belonging to this pattern 1 here were also found in clusters that were exclusively found in individuals during recovery or after recovery of COVID. So After that, we continued sample collection, and by now we have, I think, somewhere more than 70 samples of recovered patients. So we were really wondering if these expanding clones could be somehow associated with recovery from disease. So we continued this analysis, and these are now data that we just submitted for publication. But I think they're really intriguing because what they show is we have 4 cases here on the right where we have cases who had a fatal outcome after the infection with SARS-CoV-2. And then the 2 cases on the left recovered from the infection. And we see that these 2 cases on the left, one of those is patient 7, who we already showed before, have these expanding pattern, or have these clones who expand towards recovery. And we were wondering if, first of all, we can find these clones in other COVID-19 patients from whom we probably had just 1 or 2 samples, let's say, and see if we can find them during active disease or recovery, or in severe or in fatal cases. And what we did was then we extracted all the clonotypes that belong to these red trajectories. These are 176 clonotypes from these 2 patients, and around 40% of those are public. So we went then on and looked in these other repertoires of patients with COVID, and we found 13 of these clones in over 100 repertoires that we were mining at that time. And the 13 clones are shown here. And most of them, so 11 of them, are public. They have a high generation probability. And what was most interesting to us was that actually we could find these clones mainly in patients who recovered from COVID, and not in patients who had a fatal disease course. The recovering patients are shown here in blue. So since the majority of these 13 clones were public, we hypothesized that they could also be part of the immune repertoires of individuals who were not previously exposed to the virus. And to test this, we searched these clonotypes in pre-pandemic samples that we had available. And we had a cohort of about 150 healthy donors and a little bit more than 850 T-cell repertoires of patients with hematological and solid cancer. And interestingly, we could find these clones to some extent in these repertoires, as you can see here on the right plot. So nevertheless, these SARS-CoV-2 associated clonotypes to me had a really intriguing distribution pattern, because as you can see here on the bars, it was most likely that we find these clones in patients who recovered from COVID-19, and followed by patients who had a fatal disease course, as well as young healthy individuals. And cohorts who were least likely to have these clones or to harbor these clones in their repertoires were healthy above the age of 60, as well as patients with cancer, either solid or hematological cancer. And we were really interested to understand why these patients, although they had these clones, these patients with a fatal disease outcome, why they didn't survive, if these clones are really associated with recovery from COVID-19. And we looked at these repertoires specifically, and besides this patient too, we saw that these clones only appear once during the timeline of the samples that we were collecting from these patients. And it could be that they not successfully expanded these clones as, let's say, they should have to cope with the infection. And what I found really nice was last week, this publication by Tcell et al. That were actually looking at severe and critical cases with COVID using flow cytometry and really nicely profiling T cells, the functionality of the T cells. And they showed that actually the survival from COVID and the clearance of the virus were not associated with T-cell kinetics or the magnitude of the response. So in light of our data, which means that also these really critical cases have actually an active immune response, and it's not that they are not able to mount a good immune response towards this infection. But in light of our data, one might hypothesize that they're probably expanding the wrong clones or not the right clones, let's say. And with this, I already at the summary slide of my talk. So what we could see was we could find clusters of T cells that are specifically associated with the recovery from COVID-19. And we could also identify groups of clones which expand towards the recovery and might be indicative for a successful immune response towards the virus. And we could also, as Ms. Balchak also said, see that there seems to be some pre-existing SARS-CoV-2 immunity in pre-pandemic samples. And with this, I would like to really thank you for your attention and all our collaborators who made this study in such a short notice possible. And of course, we would like to thank all the patients for their sample donation.
Great, thank you so much for that. That was very interesting. So we have some questions streaming in here on the chat. So first, regarding the V usage in active versus recovered states, were you able to match donors by HLA genotype for that analysis?
Who could we match? Could you please repeat?
Were you able to, were you able to match the donors by HLA type?
No, because we had the HLA types, but the cohort is still really small to do these kind of analyses to have, let's say, a significant outcome. So no.
Okay. So I guess the asker is asking whether particular HLA overrepresentation in recovered versus active or severe might lead to differential V gene usage. So maybe your continued data collection that you were mentioning, maybe that's a question you guys can ask when you have more samples.
Yeah, I guess so. So at this time point, we didn't. With the immunity paper, we also published the HLA types of, I think, the first cohort, which was 39 patients. But at that time point, we didn't see any correlation of recovery in a certain HLA type. But to look at the V genes, we could do that, but I think it is still too little data to see a clear-cut picture there. But definitely that's an interesting one.
Mm-hmm. Okay, thank you. So another question that's come in is for cohort 2, your severe cohort. What was the median time to death post-infection? And was that pre or post expansion of some of the clones you mentioned that were also associated with recovery? And could the time to death skew the presence of the putatively protective clones?
Well, we didn't see these expanding clones. You mean the ones that we, yeah, so let's see here. Actually, I didn't look at this way. So, well, it's the patient 10 here, for example. Can you see my screen?
Yes.
So at the patient 10, we find one of these clones at the time point 5, and then after time point 12, I think the patient deceased, which is, we can look it up here, which is day 58 after onset of symptoms. And then in patient 44, it was actually day 43 after onset of symptoms. And that's the time point where the clone was there. But then in the 2 patients who recovered, we see these clones expanding already at day 27. And, okay, this is not really clear here. Let me see. Oh yeah, this patient was a really interesting one as well. So this one had an expansion of this clone at day 62. So yeah, that could be actually an interesting point to think about. So we have two cases. There's one who expands these clones really late and survives, and then one that expands these clones really early. So it could be something, yeah.
Yeah, so to that point, you were just showing slide number 9, I think. Your next slide. So these recovery-associated clones that are public, could it be that those who recover happen to have the right clones in their naive repertoire?
Yeah, I mean, it could be. It could be, and that would explain why some patients then, or why some people then react really with mild symptoms, you know. And what we see is that these clones are in healthy young, or like, yeah, in healthy individuals under the age of 60. They're more common or more probable to be there. And we saw in earlier work that we did that actually also these patients or these groups of people have a really rich repertoire. So it could well be that they just have more to choose from, you know.
Yeah. Very, very interesting. Okay. Let's see. Another question. Do you think these, in quotes, fatal clones could be associated with a secondary healthy issue of a patient?
The fatal clones.
Broad is the historical the healthy issue of the patient.
I cannot answer that. I'm sorry. I don't know.
Yeah. Yeah, no problem. If that questioner wants to add a sentence or two to clarify the question, feel free to do so. Meanwhile, I'll go to the next question. Regarding the clonal trajectories, did you see clonal groups with similar features in terms of the kinetics, but which appeared expanded earlier compared to your timeline? I would expect that there could be several waves of clonal expansions associated with recovery, which starts at different times, some earlier and some later.
Yeah, that was actually what Ms. Walczak was also showing in her study, right? So we didn't look for these kind of different patterns, but we have the data, so that's definitely something that I'm gonna write on my to-do list to look for, because we were only intrigued by actually these expanding clones towards the recovery state, where we could clinically see that this patient is actually recovered from disease.
Right. Okay, thank you. So with that, we're at time, and so we will move to the next speaker. So any additional questions for this speaker, you can bring those back to the panel session.